Okay, so I want to continue and finish this part about microscopy as it relates to staining, because we're going to do staining in lab next week. And I've got some useful slides to talk about the reasons for staining. But briefly before that, this picture is trying to show what you can see well on a light microscope. So this is a light microscope, yes. Light microscope, no. And sort of the minimum limit size is 0.2 microns. 0.2 microns. So light microscopes are not useful for every kind of visualization. So in this picture, this is light microscope, and we're looking at actually a relatively enormous eukaryotic cell here. This is big compared to what we were looking at yesterday. And you'll notice it's clear. And this one is so big that the clearness is not that much of a problem, but it's unstained and live. This one is phase contrast. And we have one of these microscopes down in the back of that lab, but we don't use it in our class. Phase contrast lets you look at a living cell when you don't want to kill it and stain it, but you still want to see features of it. You can use phase contrast. This one I don't remember what it is, but light microscopes are not useful for every type of microscopy. The other main type of microscopy that you wouldn't use a light microscope is if your thing is so small. So here's our 0.2 microns. That's the same number. Or 200 nanometers is the same measurement. Anything that's smaller than 200 nanometers, light microscope won't do so well for you. And then you need to go to electron beams. I don't have the picture of the visual spectrum here, but when we go down to blue light, that's the smallest wavelength we can see with our eyes. So then you might as well go to electron beams, if you can't use your eyes anyway. So electron microscopy is using electron beams, and there's two kinds. There is scanning, which makes 3D pictures. And that is bouncing electrons off the surface. So it's kind of like radar in concept. If you aim a beam and the electrons bounce off, and a computer then detects them and gives you an image. And it will look 3D. So this kind of picture, it's on my other slide set, hang on. My starting picture at the beginning of the semester, this one, this is a scanning EM for electron micrograph, and they put fake colors on there. It would have come out gray. But scanning EMs are my favorite because they look 3D, and if you could shrink yourself down and really look with your eyes, that's how the surfaces would look. Those are scanning. The other type of electron microscopy is transmission. And that one's abbreviated TEM, transmission electron micrograph is the picture. And transmission gives you 2D pictures that are like cross sections. So they look like a flat cross section. And this next photo, it's not a photo, this next image is a TEM of viruses, I think. And this is not the correct, this is not 0.1 millimeters. That's a wrong, the symbol didn't come over correctly when the computer processed it. It's not 0.1 millimeters, it's likely 0.1 nanometers, most likely. But you can see this picture is flat cross-section, and it looks less beautiful than an SEM, but that's TE. So you'll see a lot of pictures in the book that are electron micrographs. We don't have an electron microscope here, but they're very useful for looking at tiny structures in life. So now I want to talk more about staining, because we're going to do staining next Wednesday, and I want to talk about the why of it. So those slides that you looked at yesterday, what are the good colors for us that we look for? Purple, pink, red, blue. Those are your good colors. Those are the colors of the stains that were applied to the cells. If you did not apply stains to the cells, you will find yourself trying to look at the difference between gray and other kinds of gray. Or it's just light refraction that shows you the images. And it's possible, you can do it, but stains will greatly increase contrast. So the reason we do stains is to increase contrast of the structures and make them easier to see. To understand staining, there's a key fact that makes it all make sense. And this key fact is that cell structures have a negative charge in general. It's not that the cells overall have a charge, but the cell macromolecules tend to have negative charge, largely because they have phosphate groups on them. So, in general, big structures like cell walls and DNA, RNA, proteins, their overall tend to have a negative charge in solution. So, if you want to make a dye stick to cells, what charge does the dye need to have? Yeah, if you want the dye to stick to cells, and cells are negatively charged, the dye should be positively charged. So, if you use a positively charged dye, it's called basic from its chemical nature. Basic dye. And these are examples of basic dyes. These three we use. Those are the three that we use in this class. Crystal violet, methylene blue, safranin. Safranin is red. And that's called also a positive stain. So positive charge is a basic dye, and it's called positive stain because the cell gets the color. That's a normal kind of stain. And all the stains that we do in our lab are positive stains, and you end up with a colorful cell on a paler background. There's also a negative stain where you're not trying to stain the cell, you're trying to stain around it. That's negative stain, and everything about negative stain is backward of what we just said. So it's acidic dyes, the cell is colorless and the background gets the stain. So the dye is repelled from the cells. You might think, well, why would you not want to just look at the cell against the background? There's certain times to do that. If you have a cell that has something interesting around it, and you want to see what's around it, then you might stain the background, like a capsid stain. So in this one, that's actually the cell that I drew. Around it is a capsule, and if you do a negative stain, you can see the depth of the thickness of the capsule. And you can do negative stains with Sharpies. We might do one next week just for fun. Take a Sharpie, which is ink, which is negatively charged, and color, and then look. We'll probably do one of But the other ones that we do, we're going to do simple stain next week, gram stain next week, and those are positive stains with basic dyes. More staining terminology. If we call something a simple stain, we're using one dye. Thus, there will be one color of everything that dye binds to. We'll do one of those first. So if you've done the thing where you do a little cheek swab and you put it on a slide and you add methylene blue and BIOL, that's simple stain. One dye, one color. Anything that dye touches will be that color. What's more useful for microbiology is to do something called differential stain. So in differential stain, you have to have at least two dyes. Therefore, you're going to have at least two colors possible. And differential stains give you information about cell properties. The color that comes out on the cell will tell you something about that cell and possibly make it different color than others. So that's differential. Gram stain is differential. And this is an example of gram stains on two different cell types. This type of cell is considered gram positive because it comes out purple in this stain. And there are structural reasons for that we'll go into later. But both of these cell types went through the same staining process. These came out red, these came out purple. And that helps us classify them gram positive, gram negative. And I'd like you to memorize the colors for gram positive, gram negative. Gram positive is purple, like positive purple. Gram negative is red, like when you come to a red light, that's a negative thing. That's how I remember this. Realistically, the red is pink, but that messes up my memorization. So it's going to look that level of red, not this level of red. But I memorize it as red because it helps me keep track of them. I have a problem with when there's two options, like left and right, or north and south, or like purple and red. When there's only two things, I tend to get them mixed up. If there's four things, I don't have a problem, or three things. So we'll be doing that next week in lab. Acid fast, I'm not going to go through particularly. We looked at a spore stain yesterday. The one that we looked at was not differential, it was simple. Actually, it used to be differential, and the green, those are old, and the green dye has faded away. So now let's go and talk about some history of microbiology. Start on this slide. So in this section, I like to start by trying to get you to imagine a world without microbes. And that's silly because there has never been a world without microbes. But in people's imagination, up until about 1800, 1850, let's say, there were no microbes that people thought about ever, or knew about, or recognized their importance. So in that kind of world, when people get sick, why were people sick? What did people say? Like, you are sick because why? demons. That's actually like where I'm heading here. So if you don't know about microbes and you don't know that some microbes cause some disease, people get sick and frequently die back then for no apparent reason. So it must be like demons. That's a little darker than I usually say. Or like through the Bible, God is displeased with them. So you do not have God's favor. That must be why you're sick. And people that don't get sick, they're, you know, good with their God. And that's in several different religions. So having the recognition that there are microbes is a big deal, and that they cause a disease is a big deal, and then how to treat it, that's a big deal as well. But this is new stuff. I need to get that out of my hand. So first fact, microbiology is a relatively new area of biology. Because you can't study things if you don't even have the imagination that they are there in the first place. If you can't see it, how do you know it's there? That's the big question that relates to early microbiology. The way that what I want you to know about microbiology history is for these individuals, poor Cone, I don't talk about Cone, but everybody else on that list, I'm going to say briefly what they did and why it mattered and some facts about them. And I usually have this as like a match, maybe not in this class though. I want you to be familiar with, you know, the major names and what they did. Not what country and not so much the particular year, but what did they do and why was it important. And I think they each have their own slide coming up in this list. So we start with Hooke in the 1600s. If you go around in London, there are these blue plaques for famous people. You might just be walking down the street and they have them at relevant places. So that's Hooke's memorial plaque in London. And his claim to fame, oops, I want to use black. His claim to fame is, does anybody know what it was? What he did before other people?
First microscope, or like first to look in them?
It's not the first microscope, but the first to see cells. We think he's the first to see cells because he wrote it down and reported it. So he was using primitive microscopes, let's say, and he noticed a pattern in cork that was like that. He was looking at cork, he saw that. To him, they look like monks' cells that monks live in. And it was the cell walls of plants. And that's what he saw. He didn't quite know what it was, but he named it cells. And for that, he became famous. And he was also apparently a raging jerk. Somewhat famous in his day. We say that he's the first to see cells. And he was using something like this as his microscope. It looks like a syringe or something. That was Hooke. The next has a similar, maybe similar name, Lowenhoek. I do not make you learn to spell Lowenhoek. So if you end up writing his name, try to get close, but I don't count off for spelling. And I generally don't count off for spelling this class except for a certain question on the lab exam. So get close. I'll read it to myself. If it sounds like the word, that's how I approach spelling. Lowenhoek lived around the same time as Hooke, a little bit later. And his claim to fame is that he made huge advancements in microscope. Let's say in building microscopes. So back in those days, the lenses that they used for microscopes were spheres. Spheres like this. Not flat ones that we have now, but spheres, because a sphere will magnify as well. And Hooke's, the type of microscopes that Hooke was using, they had spherical lenses as well, just one at a time. Leeuwenhoek started using microscopes. Anybody read why he even wanted to use a microscope in the first place? This is hilarious to me every time. So he had a weaving business. He was a cloth merchant, and he needed a better way to do thread count. Like when you buy sheets and they have a high thread count because the threads are more thin and then they feel better. He wanted to look at thread count. And he made better microscopes to help him look at thread count. And then he was just a curious guy, and he kept going and started looking at other things. So we have a lot of amazing discoveries by historical figures coming up. And for all scientists, right, and then talk. I'm trying to write characterized. So people who made amazing discoveries in science, there are some big characteristics that they have. First one is that they have natural curiosity about the world. So they're interested in the world and how it works. They notice things. This is why I'm better to teach than to discover. Like they're curious about the world, they notice things, and they don't just go, huh, that's weird, and then go on. They notice things, and then they think about them, and they keep on investigating. So I'm going to put, they keep working. And then, the people that we know about and consider scientists, it's because they shared their knowledge. And all the people that we're going to talk about today in the history of microbiology are European white guys. European white guys are not the only scientists back then, but because they had a somewhat of a common language community, and because there was printing press by that time, that matters to this too. People would, Leeuwenhoek could read about other microscopes and how to make them and so on. And then when people found things, they wrote it down and other people read it, and that advanced science. So they're amazing scientists in other areas of the world too, but the writing and the language community is one reason why we tend to study European white guys, which Leeuwenhoek was. So he became famous in his lifetime too, and he had a portrait painted of him, which is like super status back then. Got a portrait, and he's holding his microscope, which looks like a door hinge or something in that view. This is one of his microscopes, and I think there are 17 that are known that he made, and they haven't been found. So I like to really point out, look how it looks. People probably have thrown them away from their attic, or there's still 17 out there. There's a little spherical lens. This is like a pinhead screw. That would be the stage. That would be the stage. And then you crank it up, crank it down. And this would be a fixed distance lens, yeah.
Since 17 were made and none of them were found. how
More
many
than-
tests
more
were
were made.
inside?
He made more than that. I think it's that 17 of them, like maybe he made 60 something. I don't know that number, but 17 stuck in my mind as they know that he made a lot more than have been accounted for now. Good question. So yeah, that is a real one that is confirmed to have come from his time. This picture was drawn by Leeuwenhoek. So when you can go into lab and you have your phone to take a picture, I hope you don't take it for granted because that's like miraculous. Another thing that scientists used to have to be able to do well was to draw. Any of you on the Italy trip where you studied Da Vinci's? Well, there it is. Like, we would have a great reduction in the number of scientists today if we all had to be able to draw like that. But you had to be able to draw so that you could communicate your stuff, and this is what he drew. Different organisms. That's the E, F, G, H. What does this CD thing mean? What does he mean by that? It's a dashity line.
Oh, it moved.
It moved. He watched this, and it moved. Whoop, it did a loop-de-loop. It moved like that. And he, just by looking, he was the first person to really look at microbes that we know of that wrote it down. And that just would have been so fantastic. He was looking into a whole new world that people didn't even know about. So even in this, he's shown there are different shapes of bacteria. They can move. Some of them organized together into groups like this. That's just in one figure that he drew. This is equivalent to this. This is a modern day picture of someone using the microscope. So this image uses his microscope. That thing gives you this view today. It's amazing. He's my favorite of all the people I'm going to talk about. And I don't talk about all of them that long, but just so exciting.
How do you use that microscope?
You put the thing that you want to look at on the tip of the screw. So getting it to stick there might be tricky. And then you twist the screw up until it comes into the focal distance of the tiny spherical lens, and then your eye is on the other side of it. So you hold it up like this. It kind of would look like an eye test or something. And some of the stuff that he drew, we think that we haven't found the really good microscopes because when people look through them, he drew things that are accurate. But either he had super amazing eyes or his best microscopes have not been found. So you would hold it up to your eye, and you would need to be probably outside in the sun to see it. So if he saw this, it would have been the tiniest blob of substance on the tip of that with the microbe going over its surface. Tiny, tiny, tiny. So great. Leeuwenhoek. That's weirdly out of place. Okay, sorry. Once in a while I will think, I want to talk about so-and-so at this point, but I already talked about that. Which one of these is light microscope, if you had to guess? A, because the cells aren't very big. Which one is scanning EM? B because it's 3D, looks like corn dogs. And then this would be transmission EM of the same kind of cell. Okay. At one point, I would have been switching slide sets. I'm going through the people in time order, not necessarily importance order. And we're making a big jump in topic here when we get to Jenner. He was also English, and there's a picture of him. This is a painting after the fact. Like they recreated, this became so famous they made a painting of it after the fact. And he's doing something to this little boy. What is he doing? He's getting inoculated. So what she said was he's getting a shot. That's what we would call it today to our children. He is getting inoculated. And what Jenner did, today we call it vaccination. Today we call it vaccination. But it was not quite getting a shot. So I want to write next to this list for Jenner how he arrived at this, because this is a cool story as well. Okay, so the first big word that we have to write for Jenner is smallpox. These were the days when smallpox ravaged the world and killed many people. So he's living in a time of smallpox. And what he noticed, because he was curious, and I guess he took an interest in people, is he noticed that cow maids, it's not a word that we have anymore, cow maids don't get it. Cow maids tend to not get smallpox as much as other people. So that's what he noticed. And that required him taking an interest in people that were like way below his station. He was a physician, which even back then is pretty prestigious. Cow maids are not prestigious. I don't really know like how he noticed that, because that's not, how do you know that? I don't know. I don't know how he knew that, but he noticed it. So that was the first thing that he noticed. And the second relevant fact that he was aware of, or that connected, is that cows and cow maids can get cowpox. So cow maids don't get smallpox. They work with cows, and the cows and the cow maids sometimes get cowpox, which is kind of similar but not deadly. And he didn't just go like, huh, I guess that's one good thing about being a cow maid. Like, that would be me, and then I would go on and do some trivial task or something like that. He connected it, and then he had an idea, or a question: is cowpox protective? Is cowpox protective against smallpox? That was his idea. And he also had the inkling that cowpox spreads through the sores. Like, they probably knew that smallpox spread through touching people that have it. And if their smallpox sores are broken open, it's easier to get it. So that knowledge was out there. So his question was, is cowpox protective against smallpox? So what he is doing here, what he is inoculating, is he scraped cowpox pus into the boy. So that instrument that he's using there, he had gotten cowpox pus, which this sounds gross, and he put it on the boy's arm, apparently, as depicted in the Let's Praise Jenner painting. And I always think, like, the boy's mom, like, who is this boy? Was he an orphan? Like, how did this all work out? That I don't know. And the boy didn't get smallpox and die. That's good. That's a sample size of 1. Jenner kept doing this, and then it eventually became somewhat accepted that this could be a way to prevent your children from dying. But even back then, this is hilarious. You can go and find cartoons that are they're called anti-inoculation. And they've got like cows in a room with people, and like they're trying to say this is stupid and gross.
It's still relevant today.
It's still relevant today. I have that picture in my non-major slides because I spent a whole day about vaccination for them because they're going to be doing it. Any of you going to do pharmacy? Maybe you? If you do pharmacy and you know, some of you do like doctor, the doctors don't vaccinate. It's too lowly. You've got more important stuff. But you would do a lot. Yeah. It's interesting. Jenner became famous in his lifetime for that, even though inoculation was not universally accepted. And it wasn't known that there's a virus that causes that. It's just like, oh, this fixes the problem and protects people. You don't have to know everything about what's going on to make a difference. Okay, now we come to the more pure research guys. So now we come to Pasteur. I'm sure I'm not saying that right. Pasteur was French, and he did a lot of important things for the field of microbiology, partly because he was motivated to improve wine and beer production in the country of France. And so that was his one of his beginning motivations to do the work that he did. Pasteur is so famous that he, at least when I made these slides, he was on the money for France with his, I don't know why he's playing with the dog. Yes, I do. Yes, I do. Rabies. He helped work on rabies problem. He's got his apparatus there. Their most famous research organization in France is called Pasteur Institute. There's still amazing biological research going on there. One of the things that Pasteur was most famous for is that he, I'm just going to put it, debunked the theory of spontaneous generation. How many of you studied this experiment in high school? That's pretty good. I'm pretty sure I didn't. Okay, so the theory, somebody want to summarize what is the theory of spontaneous generation? What does that mean?
You just randomly get sick.
It's related to that you just randomly get sick. It's very related to that. Spontaneous generation mostly came from people noticing food spoilage. Yeah, so here's the example, the way that textbooks write it. If I leave a piece of meat, if I put a steak here today, why would I do this? I would not. A raw one, and I put it here, and I look at it today, and I examine it very carefully. It's just a steak. I don't see anything else on it. If I come back a few days later, it will have maggots on it that came from nowhere. If on day 0, the first day, I look all through the steak and I look everywhere, the maggots were not deep in it on day 0. But by day 3 or 4, whatever, there are maggots all over it. They spontaneously generated. That's what it seems like, right? Where do they really come from?
Flies or dragons.
Yeah, from flies. Yeah, from flies. Why was that so hard to figure out? Because flies are everywhere back then, and it's not so obvious. So, so that was the beginning of people saying that spontaneous generation exists. Also, you know, if you let milk sit out, it's going to spoil. If you have broth, so these experiments were done with broth, and you put broth into a container, if you just leave it, it will putrefy. So if you just leave this, it will putrefy, and we don't use that word anymore, but that's what people called it back then. It will putrefy. If you take the broth and you boiled it, it will take it longer to putrefy, but it still would putrefy. So, you know, it started with the maggot concept, and then with this putrefaction of liquids that sit out. So there were two big. One big theory about this is that people knew that if you covered it and no air could get in, that it wouldn't putrefy. So if you put a cover on this after you boiled it, it wouldn't putrefy. So then people said that spontaneous generation requires a vital substance from the air. And someone named it even, it was called phlogiston, or I'm saying it wrong. There's a vital substance in the air that's required for this spontaneous generation. Pasteur and a lot of other scientists were pretty sure that was not what was happening. But what he did that was genius to prove it is he took broth and he put it in this thing called a swan neck flask. So this is indicating that you bend the glass and make the flask have this bend in it, and then you boil it, and then you let it sit. If air has the phlogiston, the vital substance, what should happen after this? It would still putrefy. So if air is causing putrefaction because it has the vital substance for spontaneous generation, this should putrefy, but that's not what happened. So Pasteur did this, and this is actually not the most useful thing that he did, but highlight it as a way to think about experiments. So Pasteur did this, the liquid cooled down, it stayed. You can go to the Pasteur Institute now and still see these sitting there not putrefied. I had a friend that got to go. Oh, I'm so dorking out. Get to see the swan neck flask, and they're oxidized, but they're not putrefied, even after 200 years or whatever it's been. Still now. So air can get in, but it's not just air touching that makes things putrefy, it's things that fall out of the air. Just like on your plate, if you leave your agar plate sitting around, things fall out of the air and it will grow microbes. Mm-hmm.
Is this where the word like pasteurization?
It does, yes.
Okay.
I was going to write it and let's put that here too. Pasteurization does come from his name.
'Cause that's
And-
like what that is, right? Like
It's very related. So what pasteurization is if you heated, let's go back to this picture. No, let's go back to this picture. No, this one. If you heated but not boiled, like just a little bit, and just kind of get it slightly uncomfortably hot for the microbes that are in there, but not boil it, and then you wait a while, it putrefies less, or it putrefies more slowly. But in the meantime, the thing that you want to eat or drink tastes better than if you had boiled the crap out of it in the beginning. Yeah. So if your grandma makes something and she talks about, I'm going to scald the milk, that's pasteurization. That's the difference between when you buy milk in a regular jug it's a ticking clock. You got a certain amount of time before it's putrefied. But if you buy shelf-stable milk, well, it can sit on the shelf for years because it's actually been more than pasteurized. So he came up with that too. And that was related to wine and beer production, that part of it. He also used what Jenner learned to help fight against rabies. That's why the dog was in the picture on the money. Did a lot of good for the history of microbiology. Oh, here's the end of it. If you either do this and get the broth to touch the material from the air and set it up right, the liquid will putrefy. Or, same thing, if you break the neck in the vertical region, it would putrefy. So he showed that also, and that was really convincing. This is a very elegant experiment. It would kind of be like, to relate it to how the public thinks, like if someone right now could do some kind of experiment and it would definitely show that vaccines do not cause autism, that would be the level of this kind of public back and forth that he kind of had the nail in the coffin. I don't think there is that kind of experiment, by the way, that we could do, because now we know about multifactorial gene-environment interactions. Anyway, next guy, Cook. It's pronounced Cook. He's German. He was a contemporary of Pasteur. And they kind of competed over like national beer production, which seems weird now, but who's the winner, do you think, and who came out on top in national beer production? France or Germany? Germany. France was ahead until Cook got started working on that problem, and France went more down the wine route. But Pasteur and Cook were started making it scientific. It was already known how to make wine and beer, but they recognized microbial action in producing it, and they started working on the science of it. But Cook's major accomplishments include figuring out how to do aseptic technique, which is the underpinning of all lab research into microbiology. So without him, he developed the rules for how do you study microbiology in a controlled setting, which is a big deal. Something else that he did I think I talk about this maybe way later in this semester, but he developed Cook's postulates. Anybody know what these are for generally? These are for how do you prove a certain organism causes a certain disease. He developed rules for that. So we take it for granted that we know that AIDS is caused by human immunodeficiency virus. You just know, you know. Like, what's another one? We just know that staph infections are caused by Staphylococcus bacteria. So he was building on the foundation that microbes exist in the first place. They might cause disease. He made rules to show that this microbe causes this particular disease. So when we discover new diseases, people still use those rules to say, oh, that disease comes from that thing. Mm-hmm.
So was he able to support that, like, rats were like a main cause of the plague, or was that already determined? Or the feces?
People had figured that. I think people had figured that out.
Okay.
Like they had seen the connection, if that makes sense. Like if you could
That's
get
like
rid of rats,
years
plagues would go down. That tells you that that it's the rats. It's not the rats, it's the microbes that the rats have that actually are on the fleas on the rats.
Yeah, so they didn't determine that until later, or was that at that time? Because this is like hundreds of years later.
It is hundreds of years later. So I don't remember like what particular diseases he started with first. It wasn't that one, because that wasn't the emergency of the time. People tend to work on what's the emergency of the time, which makes sense. For him, it wasn't plague. I think it was pneumonia by that time. But that type of thing. It's interesting to think about, like, yes, rats. Yes, rats. But the rats aren't the direct cause. Good question. So one of the things that Cook figured out was aseptic technique, and we learned to do aseptic technique next Wednesday in lab. But he figured out that there are lots of microbes around, mixed together. Lots of microbes around mixed together. And also, if you're really going to study them, you need to study them individually. Here's a silly example. Like, you know, if there was some kind of giant alien far away, and they determined that there were things here called animals, and they lived in a zoo. Animals are in the zoo. How can you study just the white-handed gibbon, if that's even a thing? Like, there's some animals over there. They're all mixed together. I want to know stuff about white-handed gibbon. I need to get it on its own, away from the tiger.